How to register a new account on the ICAR PT platform. Browse ICAR.org. Under the icon Milk Analysis, select Proficiency Test. Here you find registration to the ICAR Proficiency Test for the new users. Click the link. A registration mask appears, report your details and submit. You will receive an email from pt@icar.org when the account is activated, with the email used during the registration. In Round List, choose a round where you would like to participate. Please note that the round has a start and end date. It is possible to insert your subscription only before the run start. Click on the right icon. Now you have to subscribe the parameters of this round, where you would like to participate. Click on New Set of Test, and activate the parameter where you would like to participate. In this example, the parameters fat reference, protein reference and SCC have been chosen. We remind you that the parameter SCC includes all methods. In case you wish to order another set of sample, for example you are planning to participate with 2 instruments, click on Add Set of Tests, choose the parameters of this second sets of samples, and save. In this example the laboratory has chosen, first set, fat reference, protein reference, SCC. Second set, fat reference, protein reference, urea reference, SCC. The laboratory will receive 2 sets of fat, 2 sets of protein, 1 set of urea, 2 sets of SCC. Insert your results. After having entered into your account, the subscribed order is visible. For your internal use, you can now print the result by clicking on the blank form. When the results have been produced, you can insert them by click Insert, for each parameter that you have subscribed. Download your performance. As soon as ICAR will send you the email that the reports are published, you can log in your account to select the round, and to download your performance. You can choose the short report, with only your performance, and the complete report, with your performance and the statistical elaboration. Download your reference PT or routine PT, and you will find the contents. Introduction, where are reported updated information of the ICAR PT ISO 17043 round. Your performance analyzes. Control charts. ICAR statistical elaboration, for each parameter offered by ICAR. The introduction is standardized, but it is suggested to read each time. Here are reported the last updating news of the ICAR PT, that have been decided in the ICAR Milk Analyses Subcommittee. The participating laboratories, with at least 1 parameter, are reported in country's alphabetic order. Your performance analyses. This is one of the most important section of the report, because it summarizes in few pages your performance for the parameters subscribed. Section A reports if the laboratory reported the results for the parameters subscribed. The code reported in the statistical elaboration for each parameter. If all the samples offered have been analyzed. Section B reports if the results have been submitted on time, according the ICAR PT schedule. Section C indicates if the results have been reported in the correct unit of measurements. This point could seem superfluous, but in the real life, some laboratories do not pay attention to the unit of measurement requested by ICAR in the result sheet. In some cases it is clear, and ICAR recalculate the correct unit, but this is reported as a red flag in Section C. Section D indicates the ranking from 1% to 100%. D is the mean bias from the assigned value. SD is the standard deviation of the bias of each sample, and capital D is the Euclidean distance. D and SD are represented in Figure 1 of the statistical elaboration. The limits for the green or red icon are only indicative, and so far do not constitute standard values. They indicate what is normally reachable by labs, for their self-evaluation. ICAR Milk Analyses Subcommittee is monitoring these limits, and eventually, will update the limit of the box to evaluate the accuracy. Section E indicates the samples that resulted outlier for your participation code, for Cochrane or Grubbs test. Section F reports the absolute difference between the 2 replicates of each sample. The repeatability evaluation should be one of the first controls before any result reporting. The absolute difference between replicates is compared with the repeatability limit of the relevant reference method indicated. If one or more results have a result out of the limit, the cell is in red. Section G reports your z-score PT, calculated as ratio between bias of lab mean and assigned value and standard deviation of PT. Your z-score fix, calculated as ratio between of lab mean bias and assigned value and standard deviation of reproducibility of the method. The red cell will appear if you have a ZS minor of -3 or upper of 3. Control charts report on the graphics the ZSPT and ZSFix of the last 3 proficiency tests where your lab participated. In the associated tables are reported all the ZSPT and ZSFix where your laboratory participated. ICAR statistical elaboration for each parameter, offered by ICAR. ICAR statistical elaboration is ISO 17043 accredited, having as subcontractor Actalia PT provider. For each parameter, all the data received are presented. All the parameters offered by ICAR are reported, even if you did not subscribe the participation of some parameters. You find the statistical elaboration for all the ICAR PT parameters. Reference method report, fat, protein, lactose and urea reference method. Somatic cell microscope and fluoro-optoelectronic method. Routine methods report, fat, protein, lactose, urea and BHB infrared method. POG ELISA method. DNA PCR method. We think it is important to show you, as ICAR member, the reproducibility of the ICAR laboratories, even if you have not participated in this PT round. For each parameter, the SR standard deviation of reproducibility has been calculated after the outlier elimination. If you have participated, and your results are in the repeatability limits, you can use SR PT value for the calculation of your uncertainty of measurement.